Okay, so on opening up the simulator, I type my name in and make sure I click the BCMB3X01. Now I go into the prep room and I'm going to start assembling the solutions that I need. So the first is the vanilla. So I give it a label, choose a volume, choose the compound, click create. And I do the same sort of thing for the globin, the mutant globin, and all the other components that we need. Now, eventually I make a bit of a mistake and I realize I've labeled the tube wrongly. So I just click on the little green selector and then the tube turns red. I can change the label. Now I make a rack, and that rack's going to contain 14 tubes, and I pull the tubes apart a little bit just so that I've got a little bit more space. Notice how I can tilt and pan the view to get the best pipetting view for the next step. Notice how I can change the lids. I can put them up, open and close them. And now I'm going to use the pipette to pipette 3.5 microliters of the vanilla into each of the tubes. And obviously when you do this, it's going to be painful and very, very slow, but I've just speeded up time here. Now I'm opening the lids of the tubes that I'm going to put the globin in, and you can see when I put the globin in, their little tabs have gone red, which shows that they're not mixed. So green means mixed and red means not mixed. Now I'm going to put the mutant globin into the last 3 tubes in each sequence, and then having done that, we'll close the lids again and we'll get going with the next addition. So basically going through the additions as in the table, opening lids, closing lids as we go, changing the volumes, finally ending up with the DMEM, which I should have put in the first place, the 42 microliters of the DMEM, and eventually that all goes in and we close all the lids. And now what we're going to do is select them all, and then we're going to press the M key to mix them all. So that's a crucial little sequence. Pressing M, they all shake, deselect them, they're all nicely mixed. So that will be the end of day 1 of the prac getting those solutions. We can check the contents of them at any stage by clicking on the details button and having a look to see what's in them. It all gets a bit messy, so let's turn that off. Let's move this rack to the back and let's bring a new, we're adding the Fugene. Sorry, we're adding the Fugene, and now what we'll do with this one is we'll mix this time by pipetting up and down. You'll notice I've set the pipette to 50 odd microliters and I'm pipetting up and down to get that same mixing effect. So this is a nice thing about the simulator. We can mix either by pressing M when a tube is selected, or we can pipette up and down until the little tab goes green. There is an algorithm for that. I still haven't quite got that working perfectly. It should favor the higher volumes, of course. If you suck up and down a high volume, it should mix quicker. Now, we're going to make a plate of cells, so let's bring that plate of cells to the fore, and we're going to pipette the contents of each of those tubes into a well. Now, this, of course, is where I've set things up badly, because I've got wells which are 7 wide on the rack, and yet the plate is a 6 well by 4 well plate, so I'm bound to get confused at some stage. It's okay for the first sort of 6, and then suddenly you'll see me thinking, am I putting the right thing in the right place? Around about here, I think, is where I think, have I already just pipetted something in that one or not? Anyway, so the different mixtures are going in there. Now we're going to click incubate, and that will mimic the effect of a 24-hour incubation. Now we're going to bring the lysis buffer along, and I had to change the label there again, you see, and I'm going to put the lysis buffer into each of those, put 102 microliters that, mix, and then do the lysis. And now I'm going to take the lysed cells and put them into new tubes. I can check the constituents to see that everything's going well. There should be now luciferase in those tubes. And now what I'm going to do is make a new plate and put the contents of those tubes in and read them. So that's what we're going to do.